As you've got these three components, plasma membrane, cytoplasm, and genetic material, you do have a cell. That's the reason why viruses are not living things, because they do not have these three components. We've got the three components in prokaryotic cells and eukaryotic cells. The plasma membrane, the cytoplasm, that is the orange liquid here, and the genetic material. And we do have them in the eukaryotic cell. Then why do they have a different name? Because this one, prokaryotic cell, they only have the genetic material, but it's not enclosed, it's not inside a nucleus. It's just dispersed in the cytoplasm. Hasta ahí llegábamos todos, ¿verdad? Apart from this big difference that the genetic material is not in the nucleus, there are other differences that are not the reason for the name, but they are important differences. One is the size. Prokaryotic cells are smaller. They are more or less the size of this organelle, mitochondrion, so 10 times smaller. And there's another difference, that they are only found in bacteria, in the Monera kingdom. That's why sometimes scientists introduced another taxon. I'm not sure whether you remember Kingdom, Phylum, Class, Order, Family, Genus, and Species. Eso eran como las categorías taxonómicas, taxonomical units. Sometimes scientists include another one that is domain, como el grande, and we've got the domain in which we include the bacteria, and then the eukaryotic domain, that is the one with eukaryotic cells. Bueno, esto para el año que viene, para primero de bachillerato, lo de los dominios. So differences are that prokaryotic cells, they don't have nucleus, they are only the Monera kingdom, and they are smaller. Which one appeared first in the history of the Earth? Prokaryotic cells. And then how did the first eukaryotic cell came out from a prokaryotic cell? Well, we don't know, because there are no evidences. There are no intermediate There are no intermediate fossils, so we are not sure, but there are different theories. One theory is the one that Lynn Margulis proposed. That is that the original prokaryotic cell, the tiny eukaryotic cell that had a membrane, cytoplasm, and DNA dispersed in the cytoplasm, started folding the membrane. Do we have a nucleus already? No, not yet, just some folds in the membrane. But this DNA is protected thanks to these folds. So because the DNA, the genetic material, that is an important material in the cell, is protected, this cell is going to have a benefit against other organisms that try to damage the cell. So evolution is going to benefit, is going to choose this type of cells in specific conditions. So because this is a good trait, it's going to be passed to the following generations. And then little by little, not only this membrane will enclose the genetic material, but there will be more folds that will have different functions. They are organelles with membranes. We're not going to have those organelles with membranes in the prokaryotic cells. We only have them in eukaryotic cells. In theory, that's already a eukaryotic cell, because it already has a nucleus, it has an endoplasmic reticulum, it has more organelles. But the theory explains the following stages in evolution. Explain how this original eukaryotic cell turned into a heterotrophic eukaryotic cell able to do cellular respiration, and how it also, another branch in evolution, led to photosynthetic eukaryotic cells. What is this path? Well, first of all, this big original eukaryotic cell engulfed, se la comió, engulfed another prokaryotic cell. That was a good advanced prokaryotic cell because it was able to do chemical reactions to produce energy. So this original eukaryotic cell, instead of just dissolve it, digest it, this cell decided to start a symbiosis with this one and said, okay, I'm not going to dissolve you, but you are going to do something for me. You're going to produce some energy for me. That's how they started this symbiosis that end up at the end, millions of years later, in this ancestral heterotrophic eukaryotic cell. What are the evidences that led Margulis to this idea? Well, there are several evidences. The first one, very important, the one that led to this stage, is that prokaryotic cells do not have any organelle that is enclosed in a membrane, but eukaryotic cells, apart from the nuclear membrane that has a chemical composition that is identical to the chemical composition of the cell membrane, apart from this membrane, eukaryotic cells, they also have organelles with the same membrane, same chemical composition. So could it be possible that all of them come from the same origin? It could, it's one possibility. And then what's the other idea that led Margulis develop this theory, considering that this prokaryotic cell made a symbiosis with this original eukaryotic cell? Well, the idea is that mitochondria contain a little bit of DNA inside, and this DNA is not identical to the DNA in the cell. So they have a different origin. They must come from different organisms that one day made a symbiosis. Okay, so we've got those ideas. One is smaller, the other one is bigger, no membrane, organelles with membrane, and a little bit of DNA in mitochondria that is different from the DNA in the nucleus. And thanks to this idea, we've got this endosymbiosis theory. And then, apart from this branch, Margulis explained this other branch based on the same original idea, that not only mitochondria have DNA, but also chloroplasts, they also have some DNA that is different from the mitochondrial and is different from the nuclear DNA. So why not? They made another symbiosis with another organism that was already able to do photosynthesis. You know that the first organisms that appeared on Earth, the first prokaryotic organisms, they were not able to do photosynthesis. But later in the history of the Earth, photosynthesis appeared. So maybe another one did this symbiosis. Hasta aquí sí que habíamos visto en clase, ¿verdad? Os hago hablar para ver si estoy hablando sola. ¿Estoy hablando sola?
Sí, sí.
Esto sí lo dimos en clase.
Esto sí lo dimos en clase, ¿verdad? Vale. Bueno, so we've got to start with the components of eukaryotic cell, the nucleus, the cytoplasm, and the plasma membrane. And in the nucleus, we also saw, creo que hasta aquí también llegamos, we also saw the components of the nucleus, the nuclear membrane, the nucleoplasm, the nucleolus, and the chromatin. You also have these components in your book. Así que ahora mismo no lo tengo. In your book, you've got all that in page. You've got all the components of the nucleus in page number 13 in your book. Remember that this exam, the following exam, is going to be in 2 units. It's going to be about 2 units, part of unit 1, part of unit 2. So all this about the nucleus is in page 13 in your book. You've got the nuclear membrane, nucleoplasm, nucleolus, and chromatin. Important things: the nuclear membrane, that is the same chemical composition as the cell membrane that we've got here, but in the nucleus we've got another one. But this one is different because it's double. Double means that it probably comes from the original one that was folded. ¿Veis que es otra prueba eso también? The original membrane folded, and that's why this one is double. But even though it's double, it contains these pores that are very useful because this nucleus contains all the instructions, but the instructions must go out of the nucleus to be sent to all the organelles. So the mission of these pores is let the information going from inside to outside of the nucleus. But are they going to let DNA moving out of the nucleus? No, because the DNA molecule is very big. So what they are going to let going out is RNA, which is a copy of this DNA molecule. Vamos a ver si esto lo voy a aquí, así. DNA is a molecule made of a double strand, so it's thicker, but it's also very, very long. So when the nucleus needs to send one instruction to an organelle outside of its compartment, it's going to make a copy. That's messenger RNA. M stands for messenger. It's going to send a message. Messenger RNA that is going to go out of the nucleus through these nuclear pores and is going to be sent out. But only this tiny molecule goes out. DNA never ever goes out of the nucleus. And what are the other two components? Well, the nucleoplasm, that is just the liquid, the jelly-like substance, in the same way that we've got cytoplasm as the liquid in the cell, we've got nucleoplasm as the liquid in the nucleus. And apart from this nucleoplasm, we've got the nucleolus. Nucleolus singular, nucleoli if the cell contains more than one. In this example, it only contains one. And what's the mission of the nucleolus? It's going to form ribosomes. Ribosomes are very important this year as well. It's another organelle, it's one organelle that you've got to learn or at least have a review this year. We've got ribosomes represented as red dots here. So as you can see, they are very small, small enough to go out from the nucleus where they are formed into the cytoplasm through the nuclear pores. So nuclear pores are going to let going out just two substances, RNA, messenger RNAs, and another type of RNA that we'll see later, transfer RNA, just RNA and ribosomes. These two things go out through nuclear pores and nothing is going to get in. Okay, so my DNA is always protected inside the nucleus. Nucleolus that is forming ribosomes, membrane, pores, and the liquid. And what is this substance that we've got surrounding the nucleolus? Well, part of it is the liquid, part of it is what we call chromatin. Chromatin is DNA. Well, DNA with some proteins, but mainly my DNA. Why do I say that it contains some proteins? Because the molecule of DNA, the helix that we've got here, is folded in order to be compact, in order to occupy less space, is going to be folded thanks to some proteins. Good morning, Victor. We're just doing a review. We are with the components of the nucleus. So components of the nucleus that are aquí arriba, that are nuclear membrane, double with pores, nucleoplasm, the liquid, nucleolus, the rounded structure that is going to form ribosomes, and chromatin, that is this DNA with some proteins that help coiling this DNA so it's all organized. When the cell is working, this fiber of DNA is not going to be so coiled, no está tan enrollada, so the cell is able to read it. We call this chromatin. But when the cell is going to divide, every molecule of DNA is going to coil so much that it's going to form a chromosome. In every cell in your body, apart from the gamete, so in every somatic cell, you've got 46 chromosomes. That means 46 molecules of DNA. While in your sperm cells or ovules, you only have 23 molecules of DNA. This way, when the sperm cell joins the ovule, the zygote, that is the new organism, is going to have 46 molecules of DNA. One molecule is the same as one chromosome. But this molecule, when it's not coiled, we call this chromatin. The 46 molecules together is your chromatin. And when the cell is going to divide, it forms these chromosomes. Well, apart from the components of the nucleus, we've got to see more organelles this here that you've got in your book in pages 14 and 15. Those organelles are not all the organelles that you've got in those pages, but are just the ribosomes that you've got on the top. So you forget about mitochondria. Bueno, no hace falta que os olvidéis, pero you don't have to study them this year. And you go with ribosomes. What do you have to learn from these ribosomes? Well, they are made of RNA. So they are made in the nucleus, they are formed in the nucleolus of the nucleus, and their mission is to build proteins, very important. Where are they located? They could either be free in the cytoplasm or attached to this membrane in the rough endoplasmic reticulum. Okay, so you've got to see that that's very important. In your book, you've got ribosomes are formed by two subunits. We always represent them with just dots, siempre pintamos solo puntitos because they are very tiny. But if you could see them with an electronic microscope, you would see that they have two subunits. Esto veremos que es importante para la siguiente unidad, these two subunits. And what they do is they manufacture proteins. Well, they form proteins. Next organelle that we've got to study is endoplasmic reticulum, que le tenéis un poquito más abajo. Nos saltamos Golgi apparatus porque quiero que lo veáis en orden. Buenos días, Javier. So endoplasmic reticulum in your book in page number 14 and in this presentation that you've got in the virtual class as well. Dice, fluid-filled tubules for carrying substances. Well, the important things, they are tubules, they are made of membrane. Remember, this is one of the evidences that Margulis used for his endosymbiotic theory. Pero lo importante, rough endoplasmic reticulum, it has ribosomes that are attached, that are stuck in this organelle. Because they have ribosomes, the main mission is produce proteins. In fact, it's going to be the ribosomes that do it. We've got the explanation in the following slides. We've got here in red the ribosome, the one that is attached to the endoplasmic reticulum. Estaríamos aquí. And this is the red dot and is attached to one of these folds. ¿Vale? Así visto en grande, this is one of these folds. So the one that is producing the protein that we've got in purple is the ribosome. But this protein, instead of go to the cytoplasm, is going to be put inside the endoplasmic reticulum. Inside the endoplasmic reticulum is going to have a functional shape. So the endoplasmic reticulum is going to change the shape, it's going to fold it, and it's going to change the shape. And it could add other molecules. These green molecules, like tiny branches that we've got here, they are glue sites. So you are putting some glue sites into the protein, making it even more complex. That's the mission of the endoplasmic reticulum. It's going to store the proteins and it's going to transform them, making them a little bit more complex. And giving them their specific function, therefore. And what is it going to do with these proteins once we have a complex protein? It's going to send them to the following organelle, that is the Golgi body, the Golgi apparatus. Okay, so the protein is synthesized, then it folds, then it's packaged into these vesicles. Remember, a vesicle is just a bag, es una bolsita. When they are tiny, we call them vesicles. If they are bigger, we call them vacuoles. Pero es lo mismo, vacuola, vesícula, it's the same. It's just whether they are big or small. And they are sent to the Golgi apparatus. So we're going to see next what happens in the Golgi apparatus. In your book, you've got them the other way around, vale? Pero lo mismo la. What happens in the Golgi apparatus? Well, this one is going to receive those proteins, is going to modify them even more. It's going to receive all the components and modify them as well. Pero nosotros nos centramos en esto because this year we only care about what happens with these proteins. So it's going to receive them and it's going to form 3 types of packages: secretory vesicles, cell membrane, vesicles that will go with the cell membrane, or lysosomes. What does it depend on? It depends on what the protein is going to be used for. If the protein needs to be sent out outside of the cell, then the Golgi apparatus is going to produce secretory vesicles. If I need it for the cell membrane itself, it's going to form this type of package. And if it's needed inside, it's going to be transformed into lysosomes. Okay, so 3 destinations: outside, cell membrane, or inside. 3 different types of packages. That is the mission of the Golgi apparatus. That's why I always say that it's like the post office, because it's deciding where to send it. And in the following slides, you just have the 3 paths. ¿Vale? Este es el complejo y aquí va pasito a paso. Dice, let's say that is going to be sent, the proteins are going to be sent outside of the cell. Well, this rough endoplasmic reticulum forms the vesicle, gives the proteins to the Golgi body. The Golgi body is the one in green. And then the Golgi body produces the vesicles and sends all the proteins outside. Vale. That's one possibility. Exocytosis means exo, outside, cyto, cells. Cytoplasm is because cyto is cell. So exocytosis means you send it outside of the cell. Second possibility, pathway 2, second possibility. If the proteins are going to be needed in the membrane, in the same membrane, this is the vesicle that will be formed and the components will be part of the membrane. Third possibility is going to be used inside, and then this is a lysosome that is going to be used to digest different substances. Well, so in page number 14, you've got to study ribosomes, Golgi apparatus, and endoplasmic reticulum. Lysosomes are not so necessary. Vamos a decir directamente que no, ¿vale? Y lo he dicho, ya ha quedado grabado, así que ya no puedo echarme para atrás. So 3 organelles that you've got to study from page number 14. And what about page number 15? In page number 15, we've got just 1 organelle that is going to be important. That are the centrioles that you've got in the 4th position, centrioles. Centrioles are these, are forming these organelles. Remember that 2 centrioles with aster fibers, they form the centrosome. Esto es it's probably the most difficult thing about this part, that they have very similar names, and you're going to see that they are very similar to what we're going to see later. We've got centrioles, that are these two macaroni-shaped organelles with these fibers, these lines that are proteins, aster fibers. They form one centrosome. Okay, and what is their mission? Their mission is forming the mitotic spindle during cell division. The mitotic spindle is this structure. So what they do is they organize the chromosomes. These chromosomes already contain two chromatids, two parts that considering this is mitosis, that have identical information. So when the cell is going to divide, I need this part to go to one part of the cell, this part to go to the opposite pole of the cell. And who is going to do this? My centrosomes. They will attach to this chromatid, they will pull, so they will separate this half and that half, and then I will have half of the information here, identical information on this part, and they form a new cell. Let's see if you understand it better with the following picture. But that is the mission of the centrosome, that is made of centrioles and the aster fibers, is forming the mitotic spindle during cell division, so it ensures that every new cell contains exactly the same information. In the following one, you've got this one original cell that is going to divide. So I have, well, first of all, before dividing, obviously, it duplicates the genetic material. So for one moment, I have double information here, and then I divide it. So this new cell contains exactly the same information as this new cell. In order to ensure that the information is exactly the same, I need it to be organized not in the form of chromatin, but in the form of chromosome, having each chromosome the same information in each chromatid, in each part. Okay? Bueno, hasta aquí llegaba la primera parte. So, from page number 15, just the centrioles. Then we forget about pages 16, 17, 18, and 19 in your book, and also page number 20. Okay, from page 16 to page number 20, you don't have to study anything else. And then the last part of this unit that we've got to study is page number 21. That is, why do cells divide? The reason why they divide depends on the type of organism that is doing that. In the case of unicellular organisms, of course, when the cell duplicates, what is the organism doing? Well, I don't have one organism anymore, but I've got two. So the organism is reproducing. If this was an organism, when this cell duplicates, I've got two organisms. So it's performing reproduction. But you are also doing cellular division in your body every second in your life. Are you reproducing every second in your life? No. What you are doing is you are restoring, you are replacing cells that are old, or you are probably just growing, you are increasing your size. So that is the consequence of cell division. Why do you do cell division? If you're unicellular, to reproduce. If you are multicellular, to increase your size or replace your cells. That's what you have in page number 21. And what does cellular division includes? Duplicating DNA and dividing the cytoplasm. You don't duplicate the cytoplasm, you just divide it. Well, another thing that you've got in your virtual class, como veis, aquí sigue la cosa cargándose y no hay forma. Parece que esto funciona un poquito mejor. Bueno, no sé. Sí. Another thing that you've got in your virtual class, if we've got here, you've got at the beginning all the contents that we're going to see in this unit. So you can see prokaryotic cells versus eukaryotic cells. Eso lo hemos visto hoy. A cell's nucleus and its function as well. Coordination between the nucleus, the rough endoplasmic reticulum and the ribosome. The function of the centrosome and how and why do cells divide. All of this is what we've seen today, what we've had the review about, and it's all included in your Unit 1 in your book. And then what we've got to see is the second part, chromosomes, their structure, number, and some abnormalities, and the process of cell division. Where is it in your virtual class? Well, the first part, the PowerPoint presentation that we've seen today, is here, cells, the building block of life, including that picture. And the second part is this file. This file, que bueno, por si acaso no soy capaz yo de abrirlo ahí, es este. Este es bueno. From this file, at the beginning you've got all this theory that we've seen. Está bastante ampliada. Esto no hace falta todo, ¿vale? Solo lo que hemos visto hoy en la clase de hoy, ¿vale? Esto está bastante ampliado por si al caso alguno, pues, no puede dormir estos días y quiere leer un poquito más. And the following theory, we've got it from page number, estaría bien que hubiera aquí. From page number 4, okay? And then you've got some activities. Ahora es cuando os toca a vosotros decidme, ¿qué ejercicios os pedí? ¿Os he pedido alguno ya de estos? ¿Hay alguien ahí o estoy hablando sola otra vez?
No, o sea, no sé.
No sabes, no crees que no os he pedido ninguno, ¿no? Puede ser que no os haya.
No
Vale.
pediste nada.
Bueno, pero habéis...
cosa,
Dime.
Yo
Si
creo que
tienes-
sí.
Bueno, es igual, sí, tampoco pasa nada, no hace falta confesar
Creo
ahora.
que los tres primeros.
¿Verdad? Eso pensaba yo también, pero no pasa nada, porque igual que yo me echo la tonta, pues tú también y ya está, no pasa nada. Bueno, los tres primeros, porque son muy sencillitos y son de lo que hemos visto ahora mismo. Es igual, lo podemos corregir en estos 3 minutos que nos quedan, salvo que Andrea quería decirnos algo. Dime.
Sí, a
Dime.
ver, que si nos estudiamos de las fotocopias está bien, ¿no?
Sí, lo que pasa es que el principio de esta fotocopia, este principio viene muchísimo más. Mira, si empiezas a mirar, al principio vale, pero por ejemplo aquí te dice the function of capsule, que yo este año no os he dado nada, the function of the cell wall, dice aquí pili, flagella, o sea que en lo que es esta parte viene más de lo que necesitáis.
Pero creo que no sale lo de el aparato ese que hace que se separen las células. Como que sale muy después, pero no sale lo que lo hace.
Aquí no sale. Aquí no tiene que salir, tendría que salir aquí. Y dices que aquí, centrioles, este es. Lo que pasa es que en vez de centrosome, que es esto de los términos, pone centrioles, two bodies in animal cells which are vital to cell division as they help organizing the chromosomes during cell division. Bueno,
como salía debajo de lo de las plantas, pensaba que era ya parte de lo de las plantas.
no, esto es cómo son o sea, por qué son diferentes. Plant cells are different from animal cells. ¿Por qué? Plant cells have cell wall and chloroplast, eso es lo suyo, and a large vacuole, y animal cells have centrioles and lysosomes, que esto no lo vas a encontrar en plant cells. Esto es solo plant cell, esto es solo animal cells. Vale, igual está un poco mezclado.
Entonces sería estudiarnos a partir del punto 3, ¿no? Porque el
Claro.
punto 1 y 2
Sí,
son...
el punto 1 y 2 es para que, no sé, por si lo quieres tener más imágenes, y es que ya lo tenía todo junto y digo, ¿para qué se lo voy a borrar? Si lo quieren tener, pues lo tienen. Pero lo suyo es estudiarlo a través del libro o de la presentación, la primera parte, ¿vale? Y luego la segunda parte, lo que en el aula, ahora ya no quiere, lo que en el aula viene como separado los dos últimos puntos, esto sí es a partir de aquí, ¿vale? Que decía los cromosomas, la estructura del cromosoma, el número y las anomalías cromosómicas, que sería esto de aquí, y luego cómo se hace la división, que primero te pone la de la procariota y después te pone la de la eucariota, la mitosis y la meiosis. Eso sería la segunda parte. Los dos puntos esos, ¿vale? O sea que esto prácticamente eso, lo que comentabas, a partir del punto 4 sería por donde va la teoría aquí. La presentación para la primera parte y eso para la segunda, ¿vale? Bueno, me suena el timbre, aunque no tengo timbre. Así que aquellos que no habéis hecho esos ejercicios 1, 2 y 3, porque a lo mejor no los mandé, pues el 1, 2 y 3 empezamos mañana corrigiendo esos, ¿vale? El próximo día, ¿vale?